Hello, everyone, and welcome to the next webinar in our series hosted by the National Cooperative Soil Survey Research Committee. This is the first of a series within that focused on new technologies in soil survey. Look for some upcoming webinars on MIR use in soil survey, techniques to capture existing data, and then future webinars in this series will include a Pedon to landscape scale series coming up early next year. So as we get started with this webinar, we're very excited to offer what I think is it's really interesting work that's going on from the building block side of things. And we'll let Dan and our speakers tell you a little bit more about that. But I do want to mention, make sure that you use the Q&A pod if you have questions during the presentation. Be sure that you are muted so that we don't hear any of your background noise. You do have the ability to come off mute as we have a Q&A session after each speaker. And then we will try to answer things as things are going on and then bring it back up. But I would like to turn it over to Daniel Hirmas, who is a professor of Pedology, is that official title? I'll let him tell you, at Texas Tech University and the organizer of this webinar in our series. Take it away, Dan.
Okay, thanks, Skye. So welcome everybody to the second NCSS research webinar being organized by subgroups within the larger NCSS research committee. As Skye mentioned, my name is Daniel Hirmas. I'm a podologist at Texas Tech, and I serve as a member of the Topical Sessions Working Group on New Technologies in Soil Survey. Now, other members of the working group are Skye Wills, Meyer Bond, Yakun Zhang, Mark Kimsey, and Matt Levi. The focus of our webinar today is on exploring the potential application of new technologies to advance soil survey, especially at the micro to Pedon scales. We're really hoping that this will spur ideas and collaborations going forward in research, especially research related to soil survey. We're excited that 136 folks have registered as of this morning, which I think shows a fantastic interest and turnout. So kudos to everybody, and thanks and kudos to our speakers too, who generated that interest. Before introducing our two speakers for today's event, just a few logistics that Skye also mentioned. So the speakers each have 30 minutes for their talk, and there'll be roughly around 15 minutes for the Q&A. If you have a question, put it into the Q&A. Just be aware that there is both a chat and a Q&A. And you're going to want to put it into the Q&A if you can, but if it goes into the chat, we're also going to monitor that, but try to put it in the Q&A if you can. One of the organizers, myself, or some of the other people on the committee will help moderate and introduce questions. What we'll do is we'll call, we'll see who gave the question, and we'll ask them to unmute, and that way you can ask your question when you're called on, or we can read it from you if you prefer. Okay, so with that out of the way, our first speaker is Dr. Rebecca Lybrand. Rebecca is an associate professor in the Department of Land, Air, and Water Resources at UC Davis. Rebecca earned a BA in biology from Whittier College, MS in soil and water science from UC Riverside, and a PhD in soil and water and environmental science from the University of Arizona. Rebecca started as an assistant professor at Oregon State University in 2015 before moving her program to UC Davis in 2020. She specializes in pedology, soil mineralogy, and the study of soil landscape relationships under contrasting environmental conditions. Now, Rebecca has several graduate students online to help with the Q&A session, and they will be introduced in her presentation. So with that out of the way, Rebecca, feel free to take it over.
All right, thank you so much. And I'm hearing that we may have a little bit of issues with some lag. So I'm waving to you all and saying hi, but I am going to turn off my camera for the presentation, and I'll turn it back on for Q&A. So hello, my name is Rebecca Lybrand. I'm an associate professor in the Department of Land, Air, and Water Resources at UC Davis, and I am thrilled for the opportunity to speak as part of your webinar series. So thank you for the invitation, and I'm really looking forward to some ongoing conversations and potential collaborations, thinking about linking high resolution analyses or microscale capabilities to soil survey and having some discussions around that. The title of my presentation today is From Soil Pits to High Resolution Imaging, a cross-scale approach to understanding the building blocks of soil. And before I get started today, I do want to say that there are 3 graduate students online, Cassie, Kylie, and Maddie. And so I'm actually going to be referencing their different research projects as part of our ongoing work. And so if you have questions, especially in the ongoing work section, feel free to put them into chat, even during my presentation. And one of the students could likely respond to you, especially if it's specifics on their project. And also, before I jump into the actual research, I want to just take a moment to acknowledge all of the people who made this work possible. And so when you hear about the Mineral Mesh Bag Project or our work in the Sonoran Desert.
Rebecca?
Yeah.
We are just seeing a black screen.
Oh, okay. Let me try resharing. Did you see the title slide or anything?
No, I didn't. I don't think I saw anything.
Oh, that's great. Okay, let me hold on 1 second.
Perfect. We can see it now.
Excellent.
Okay, and you can hear me?
Yep.
Okay, perfect. All right, well, now you're seeing my title slide coming up. So again, this presentation is going to focus on a cross-scale approach to understanding the building blocks of soil. And as I get started on the presentation, I just want to highlight that when you're hearing about the field work, you're hearing about the Mesh Bag Project or any of these other components, there are all kinds of people, students, professional government scientists, as well as faculty at institutions across the US, from Alaska to South Carolina to Arizona, that have made this work possible. So I just want to fully acknowledge everyone who has made this research happen, both in the field and then also at the national lab level. So thinking about the national lab scientists, especially at the Pacific Northwest National Lab, you'll be hearing me reference PNNL and some of the different capabilities throughout this presentation as well. And it's an area of research that I'm really excited about, being able to connect soils and what we can learn about soils in the field to taking samples to analyze using some of these advanced capabilities in the national lab system. And I think there is the opportunity for a lot of collaboration. And I know specifically a lot of the national lab scientists are always looking for collaborations with those folks who are working on soils in the field. So overall, today's objective is to focus on the cross-scale analyses of soil. And we're specifically going to be looking at a few different techniques. And then I will be wrapping up with thinking about this quantitative field photography project that we're just getting going through a funded proposal with the NRCS. So when we think about soils, we are thinking about soils on the ground in terms of ag landscapes, agroforestry. There's all kinds of different applications for learning and studying soils in the field. I also always like to give a shout out to thinking about soils as a habitat and the importance of different types of soil properties from a conservation effort standpoint as well. But for the focus of today's presentation, we are thinking about connections between what we can learn for soils in the field, and then thinking about microscale types of analyses. And one of the things that our lab is also really excited about is thinking about the applications of soils to other planetary surfaces. And while this isn't a focal point of today's presentation, I do just want to encourage you to think about the applications of these different analytical techniques and of soil science across all types of planetary surfaces. For today's presentation, though, we are focused here on Earth, and we will specifically be thinking about connecting field-based soil analyses to those in the laboratory system, especially high-resolution imaging. So one of the overarching questions driving research within our program is, how do we disentangle the heterogeneity of abiotic and biotic processes that transform minerals and release life-supporting nutrients to soil? We are specifically focused at the nanotime microscale, where mineral-water microbe interactions occur. And within this context, there's a lot of emphasis on understanding the role of mineral weathering and abiotic and biotic processes, especially from a nutrient cycling standpoint, and what types of controls and drivers there are that constrain the cycling of nutrients in soil, especially those coming from rock-derived processes. So that's something that our lab is really excited about. And today you're going to hear a lot about our work on the biotic processes side, and specifically thinking about fungi and what types of techniques we can use to image fungal grain interactions. So this work really kicked off during my time in Arizona, and we did a lot of cross-scale soil biogeochemistry research, specifically focused along an environmental gradient in the Santa Catalina Mountains. So this is part of the Santa Catalina Critical Zone Observatory, where we were comparing soil formation and development from the Sonoran Desert system to a subhumid mixed conifer forest. And we were employing all of the fun techniques, so getting to go out and dig pits and take morphological observations, and then analyzing bulk soil samples using X-ray fluorescence or X-ray diffraction, and thinking about elemental loss or mineralogical distribution across landscape positions, as well as along these different ecosystems. And within this context, we also thought about carbon distribution as well as carbon persistence. But one of the components that I was especially intrigued by was actually getting to work at the microscale for the first time. And so we actually employed electron microprobe analysis to generate quantitative elemental data to better understand the distribution of elements at the microscale. So this was the first time we had the chance to connect bulk soil analyses when we're looking at, elemental loss in bulk soils to elemental loss or distribution at the microscale across different types of grains. But even within this context, one of the questions that kept coming up was, okay, we know that the grains in the mixed conifer site are showing a higher degree of transformation. Is this biotic? Is this abiotic due to the wetter climate, or a combination? And how do you get at these types of questions with respect to the microscale drivers of mineral transformation? And this led us to expand our work up to the coastal temperate rainforest in Southeast Alaska. I was especially drawn to this landscape due to the phenomenal development of spodosols and the soils overall over just a couple hundred years, due to the intense weathering regime resulting from the very wet climate, combined with the productive rainforest systems there. And so we have had an amazing time thinking about soil formation and soil development across different types of lithologies in this system. Now, within that context, we've also had the opportunity to employ this cross-scale analysis approach. We had a graduate student, Jennifer Fedenko, who went out with our team and dug 20 soil pits in pretty hard to reach terrain. So this was a big deal, just getting the soil pits dug in this environment, thinking about profile development and what that looks like, and then also bringing in the carbon side. So we were able to compare carbon distribution across this lithosequence, and then we were able to think about this between what carbon is accumulating in the subsurface compared to the overarching organic horizons. Overall, we saw that the bioclimate factors were the strongest drivers, as you could expect, and we saw relatively consistent relationships with respect to carbon distribution as well as soil development along the lithosequence that we were studying. Now, we also had another student come on who was specifically focused on carbon persistence, and we employed a density separation technique to be able to better understand what this looked like across 2 different rock types or soil formation across these 2 lithologies. And so Julia's work was really exciting because we had the density separations, the free light, the occluded, as well as the mineral fractions. And then through this collaboration with the Pacific Northwest National Lab, we were also able to image these using the scanning electron microscope, where we were able to understand differences in the complexity of the grain surfaces for the mineral fraction, what the different types of organic coatings look like. And one feature that came up over and over and over for us was the distribution of fungi throughout all of the different fractions, and seeing evidence of fungal contributions to aggregation, even in some of those occluded fractions. And so you can kind of see some examples of this being labeled throughout some of the different organic fractions that we were looking at. This again just led to the ongoing question of how can we differentiate the fungal contributors to here, thinking about the distribution of these different density separates, but also fungal contributions to mineral weathering and soil formation. So that led to this, again, ongoing question. And it's one of the points that inspired us to start with this in-soil mesh bag technique. And the overall idea with this in-soil mesh bag technique was to put out fresh sources of granular rock substrate into the surface soils, and then retrieve those. And for our project, we retrieved them after 6 months, 1 year, 3 years, and even 6 years in different components of the study. And so you'll hear me referencing some of the data sets like, oh, these are mesh bags that were deployed for 3 years or 6 years, and so on. With respect to the specific landscapes, all of the landscapes we deployed these mesh bags on were derived from granitic terrain. So one of the rock types that we put out was a fresh, coarse grained source of granite material. We also put out quartz sand. This served as our in situ control. So we wanted to understand what may be moving into and out of the mesh bags. And if we put some of the quartz sand bags on the microscope and saw iron coatings throughout, we know that wasn't coming from the rock material itself because it's quartz. So it has served as an excellent reference and background control for us to have the quartz bags out in the landscapes as well. And then since we were interested in microbial activity specifically, we did put out basalt as we called it, kind of the bait, and it was a fresh, more susceptible to weathering and fine grained material, especially that basaltic glass. It's got phosphorus and other types of nutrients. So we were, again, putting out basalt. And again, we were interested in basalt too, given all of the interest in enhanced weathering projects as well. So putting out basalt across some of these different ecosystems was also an interest due to that area of research. So when you hear me referencing the results coming up for some of the different projects, including the student-led projects, the climate gradient that we encompassed here included that Sonoran Desert site in Arizona, a humid mixed hardwood site in South Carolina. This is the Calhoun Critical Zone Observatory, and then a coastal temperate rainforest site in Southeast Alaska. Now, with us just getting going on our quantitative field photography project, that is also going to be a comparison from our coastal rainforest site in Southeast Alaska, as well as the coast redwoods sites that we're just getting going in support of that project. So when I'm referencing the field photography technique, you can also envision a nice star here. We're still establishing sites, so I don't have a specific location to share with you, but that is also on our radar. You'll be hearing me referencing Cassie's work for the SEM. So a lot of that was based in the coastal rainforest. And then Maddie's work with the micro X-ray computed tomography is based across all 3 of these environments. So she's been doing some great comparisons of those samples that we retrieved. So overall, some of the results that we've collected across the board for deploying these mesh bags and retrieving them and analyzing them across all of these sites have been absolutely fascinating because we detected evidence for biomechanical weathering, so fungal-driven biomechanical weathering in all of the sites from the Sonoran Desert to the Alaska rainforest, regardless of whether those bags were out for 6 months, 1 year, 3 years, or 6 years. It was absolutely incredible to see the degree of fungal grain contacts that happened over relatively short timeframes. And so we've got all kinds of different images that we can share from this, but these are just a handful of different examples. And Cassie can speak even more to this, given the emphasis on her project. But some of the fungal grain interactions that we saw were fungal structures actually growing through the surface mineral layer. So this is, you can see the fungal structure here growing through the mineral surface layer on this particular sample. We also saw a lot of fungal growth along the edges of the grains. And then we've seen even more disrupting the edges of those mineral layers. And so I just absolutely love these figures for demonstrating the importance of the biotic contributions to weathering. So you can see the fungi that's grown through here, and it's growing through by, again, applying that biomechanical stress to the edges of these mineral layers. And this is over relatively short multi-year timeframes. So this is from some of our initial work. And currently, and I'll be getting to some of Cassie's work more later in the presentation, Cassie is doing more replication on this type of analysis and developing some more focused classifications for fungal adherence to grains, because it does differ quite a bit. And so that's something Cassie could definitely speak to. The other important component that I wanted to include here for us thinking about techniques and microscale analyses is the fact that you don't necessarily have to use just one type of high resolution advanced capability. And so one of the techniques that we developed and demonstrated was actually how to co-locate micromineral interfaces across different types of microscopy approaches. And so this particular approach involved us identifying fungal grain interfaces using helium ion microscopy, and then identifying that same exact fungal grain interface where we could actually collect chemical information using scanning electron microscopy. And so this was a win-win. We were able to use helium ion microscopy to capture these beautiful images, especially of biological samples. And then we were able to go back to that same interface, which caught our attention here really quick. So if you draw your attention to where my cursor is in this white arrow, we saw this fungal structure, but we were saying, what is this material that's adhered to the fungal structure? And then also what grains are associated with this contact here? And so the helium ion microscope gives you this phenomenal energy, or this phenomenal imagery, but not the chemical information. But going to that same interface with the scanning electron microscope, we were able to see that this fungal structure is associated with biotite, and this aggregated material actually did not have any kind of organic compound. We thought this may have just been organic materials associated with the fungal structure. Instead, it was actually mostly iron as well as some titanium. So that leads to additional questions of, is this some kind of biomineralization process, or what's going on here? But this just provides a lot of insight. And being able to co-locate using different approaches is highly effective for getting as much information as possible. Now, we expanded this even more to investigate a fungal grain interface where we also included a focused ion beam, or FiB, to be able to open up a vertical cross-section across a fungal grain interface. And then we also employ transmission electron microscopy to identify what we're looking at with respect to these different types of mineral inclusions. And so this particular research expanded to include these techniques, again, at the same exact interface, and we were able to detect, if you picture this as a vertical cross-section, so this is where the fungal growth was, this was the contact with the underlying grain, and you can actually kind of see this dip here. So this is actually some evidence for a direct biological weathering by the fungi, that dip in the grain surface. And then we were also able to, again, use TEM, or transmission electron microscopy, to identify what these inclusions are. And we actually saw fungal contacts at several areas that had these inclusions. And these are actually magnetite grains, and it's different forms. So you can see the different morphological structures here compared to the sample, or the inclusions over here. So this particular study really demonstrated the heterogeneity of processes occurring at the microscale. And through this collaboration with PNL, we were actually able to use several different technologies at the same interface, where we were able to, again, look at this direct contact between the fungal grain interface, and also understand abiotic enrichment going on between the magnetite and the volcanic glass matrix that you see here, using an atomic probe tomography, or APT. And we were also able to think more about biotic aggregation and the influence that biota are having at this particular interface. And then even thinking about the fact that interactions like this are exposing these inclusions, these underlying inclusions of magnetite. So this could be, there's fungal mining or fungal interactions going on, and then it exposes magnetite, which could lead to subsequent redox reactions is one type of example. And then finally, thinking about just overall hydrolysis and different types of mineral weathering reactions that can be going on at the system. So this is some of our prior published work thinking about combining, samples that you can deploy in the field, collect from the soil system across climates, and then you can employ all kinds of different microscopy techniques. And the thing I want to emphasize here is there are ways to be able to move across with the same sample, move across a few different imaging approaches as well to gather more information and data about the system that you're trying to learn about. So that leads to the second part of my presentation, which I am absolutely thrilled about because we are focused on student-led research. And overall, our ongoing work is, again, still focused on the soil system, what we can learn about going out, collecting soils across these different climates, or really focusing in on one particular ecosystem overall. And so the next couple of slides are going to be focused on scanning electron microscopy and how Cassie Collins is expanding our work using the SEM to think more detailed about fungal grain interactions and associations. And then I will move on to work being led by Maddie Morris. And Maddie's work is focused on micro X-ray computed tomography of both the mesh bag samples that we've been deploying and retrieving. And Maddie is also starting to kick off some work on aggregates that are collected at these different ecosystems. And then finally, Kylie is leading the charge on developing this quantitative field photography technique that I'm just going to introduce to you. That is our latest or newest project. So we're just getting going on it. And we cannot wait to have more conversations with you all about this approach and potential applications. So if we think about the scanning electron microscopy approach and some of the ongoing research questions that we have, we are still really focused on exploring these fungal grain interactions and distribution across the different rock types, just given the importance that fungi appear to play within the weathering process and how little we still know about how this works out in field systems. There's a lot of lab-based research, and it's much harder to constrain this in the field, which is why we've been using, again, these mineral mesh bags to be able to get at this in some more detail. And so overall, since this talk is focused on these different types of approaches, the SEM, scanning electron microscopy, soil scientists have been using for a long time. We know and value SEM, but I just want to emphasize that it is also a powerful tool for assessing in situ biological contributions, especially looking at mineral weathering or incipient weathering processes. And so this is Cassie. Cassie is online and can answer any questions about this research. And I've included a copy of Cassie's most recent poster presentation at SSSA. So the work that you're about to be hearing about is actually from mesh bags that were retrieved from the coastal temperate rainforest in Alaska following 3 years of deployment. And again, this is looking at those granite, basalt, and quartz sand types of samples. And I always have to include the beautiful landscape photos in Alaska, especially when the sun is out, because that can be relatively rare depending on the season. So overall, the primary questions driving Cassie's PhD research is how does rock type influence fungal colonization? And then do specific rock-bound minerals promote a greater degree of fungal grain interaction? And this question especially is so exciting to me because yes, we can identify and say, yes, there are fungal grain contacts. Cassie is really pushing the envelope here by classifying out what types of fungal adherents do we see and how do they differ? And then also what minerals are the fungi adhere to within these specific samples? So in a granite sample, are there more fungi adhered to biotite mica compared to feldspar? These are the types of questions that Cassie is looking to address with that chapter of her PhD. And while we don't have time to get into all of the exciting specifics of Cassie's research and some of the initial outcomes, Overall, Cassie has detected fungal hyphae present in all of the samples analyzed, especially in the forms of biomechanical fungal weathering and coatings that are being observed, especially since this is coastal temperate rainforest, wetter system, kind of more movement going on with these types of coatings. And so the fungal adherence is widespread, and Cassie is observing differences as a function of rock type with respect to what these different types of interactions look like. And so one of the main takeaway points so far is that some of the preliminary results are suggesting differences in fungal distribution and fungal grain associations based on rock type, so based on comparisons between quartz, granite, and basalt, and within mineral types within some of these specific rock samples that we've been analyzing. And so again, Cassie can speak more to this, but these are some of the different types of fungal grain associations that she's been observing throughout and seeing how they differ between like a quartz sample compared to something like basalt or granite. So really exciting work. And it's really amazing to have a higher degree of replication. A lot of those papers that I was just presenting to you, they're based on a handful of grains. And you always have to ask, okay, yes, these 5 grains showed this, but what about the whole rest of the sample? And so it's been really great for Cassie to increase our replication on this a bit more. And there's still a lot to be done on the automated machine learning process, which is, again, something Cassie's interested in. But I just wanted to highlight that here is like we are thinking forward on how all of these images and data could be put into a more automated type of context. All right. We are now transitioning over. So we've talked about the extended use of scanning electron microscopy, especially for thinking about fungal grain interactions. And now we're going to think about both the analysis of these mesh bags, but also the analysis of actual soil material or PEDs that are collected in the field by research being led by Maddie Morris. And again, this is a copy of her recent poster presentation at SSSA as well. And so overall, we initially got really excited about micro X-ray computed tomography, or XCT, because it's a non-destructive analysis technique. And in a perfect world, we would be able to go to the, deploy this mesh bag, retrieve it, and scan it non-destructively and understand the distribution of grains or even the biological inputs within that bag. And this is proving to be very challenging, as you can imagine, for a bulk sample like this. But we are making incredible progress on analyzing subsamples of this material, which Maddie can definitely speak to. So overall, the goal of Maddie's research is to examine the impact of bioclimate and rock type on incipient mineral weathering of these substrates. And then we're also trying to do a little bit on the visualizing soil fungi side using XCT as well. So we've got some preliminary findings on some of those attempts. So overall, since we are talking about techniques, the micro XCT technique drew us in because this is a non-destructive 3-dimensional imaging technique, and it's based on density-based differentiation of features within your sample. And so this leads to, these are advantages. This density-based differentiation leads to advantages, but also some limitations depending on what you're specifically trying to image within a particular sample. And Maddie can speak more to the method setup here, but these are some examples of what it looks like to prep and scan some of the samples. So it's minimal sample prep. Just getting the samples into sample holders is what's needed for some of these scans. And overall, this is what we're looking at with respect to analyzing the morphology or the shape of the grains that have been deployed. So if you can imagine what one of our main goals is here is we've got control mesh bags. So we've got control basalt, for example, that's been sitting in the lab for 6 years. And then Maddie has retrieved all of the samples that were deployed across our field sites for 6 years. And we are looking to detect microscale differences in grain morphology that could point to some evidence of incipient weathering. And so in the lab, the samples haven't been subjected to any smoothing processes or anything that you may experience or that they may experience in the field. And so we were looking to test the hypothesis that we may be able to use some of these XCT parameters to detect differences between the lab control grains or samples and those that have been deployed in the field, especially under wetter conditions. And so overall, the takeaway is that XCT was employed to extract quantitative morphology data on the grains following 6 years of deployment across our field sites. And again, this could be an entire presentation on its own as well, but Maddie has explored multiple different types of shape and surface parameters to be able to analyze and to compare across the different samples. And so I included one example here with respect to surface irregularity, or this proxy. And so the idea is that, a lab controlled sample, fresh rock material, would have likely more surface irregularities compared to a sample that's been exposed to incipient weathering processes in the field that may have less irregularities after 6 years of deployment in a wetter environment or ecosystem. And so this again is kind of hot off the press data, and it's data that Maddie's including with all kinds of other figures in her upcoming manuscript. But the thing I wanted to point out to you is we've got surface irregularity here on the y-axis and the sites on the x-axis, and the black represents the control samples. And so the control samples for basalt here, granite, and quartz, the control samples for the basalt actually showed a greater degree of surface irregularity compared to the wetter ecosystems, specifically in Alaska and South Carolina. This was more detectable in the basalt, which would align with the idea that these grains may be more susceptible to incipient weathering compared to coarser grained materials such as granite. So this is just one example of what one of the output data sets can look like using this technique, but and Maddie could speak to this more. There are all kinds of different applications here and features and types of data that you can collect. The other component that I just had to include, since we are talking a lot about soil fungi, is we have been, again, really focused on different ways to visualize and understand soil fungal distribution, whether that be using compound microscopy or even some examples using the XCT, as you can see in these images here. And so Maddie has also, while imaging and analyzing these grains, has also been trying to visualize some of the soil fungi using a few different types of XCT instrumentation available at UC Davis. Another collaborator at SIGRE was able to do some trial runs for us as well. Working on this at PNNL. Again, you can see the fungal distribution, but there's a lot of artifacts that you can kind of see as like the blue, more circular shape. So it's, again, not perfect in being able to pick out just the fungal distribution here. And the latest update, and again, hot off the press because Maddie just returned from Argonne relatively recently and just got access to some of the data. She is looking to employ this to analyze some of our samples using synchrotron micro XCT because it gives a higher resolution, fewer artifacts, as well as a shorter scan duration. And so this is something that I would encourage you all to think about. Argonne, you could submit a user proposal to try to go, be able to go visit the lab and run some of your own samples. And so this has been really exciting. And while we don't have the data yet, it's really amazing to think about how you could apply this type of technique for even a soil aggregate. So I just had to include this one. This is a soil aggregate from old growth forest up in Alaska. And this is an example of one of the high resolution cross sections that you can collect from this. And so this isn't anything on the outside with respect to some of the roots that you may be able to spot here. This is an idea, a vertical slice through, and we just had to include it given that beautiful distribution that you can see of the particulate organic matter that I'm circling with my cursor. And then you can see all of the different, the porosity here that can be calculated, or even the connected porosity that could be calculated when you combine all these slices and think about the porosity of this aggregate material itself. So Maddie's continuing with this work, and we're really interested in connecting this with some of the aspects of the field photography technique, or even other types of samples that we could collect from the field and analyze directly in the lab, and trying to have things as non-destructive as possible to give a more three-dimensional view. We're really drawn to that aspect of using micro XCT for that reason. All right. Last but not least, we are going to just cover for a couple of slides here some of our ongoing research. This is being led by Kylie, and Cassie Collins is also involved for the cross-site comparison. But the main goal here is to develop a quantitative field photography technique. And this is through a proposal with the NRCS. And the whole idea here is that we can see the biological contributions to, weathering or at least soil development and aggregation out in the field. But what does that mean? And how can we actually connect observations that we make in the field to what we can see in the lab? And so the goal of this particular project is to document and assess root fungi systems in the field that could serve as biological indicators of soil health. And so how can we actually collect standardized photographs in the field of biological interactions? And what does that mean with respect to soil health? And is there any way to set this up or to connect it to any of these other types of microscale techniques? So can we take an aggregate, for example, that is photographed in the field and then analyze it using micro XCT, for example, and what can be learned from that with respect to this biological indication of soil health? So that's the overarching goal. And we are focused specifically in the coastal temperate rainforest gradient. So this Southeast Alaska site, as well as the sites that we're just getting going in the Coast Redwoods in Northern California. And we are, again, absolutely thrilled about this research. As you can imagine, sitting down to develop a field photography technique using materials that soil scientists would actually want to carry in their packs out to the field, miles and miles and miles, on top of everything else they have to carry. This is a complicated project to even kick off the ground, right? Because you're thinking about what image capture devices should we have? Clip-on lenses. What about the stands and attachments? What's not going to be too bulky? What will work well? What about the scale and background? What about lighting and color correction? What about weather and dust protection, right? These are just a few of the different conversations and discussion points that Kylie and Cassie have been sorting through to kick off this project. But it's been really exciting to think about. And the different devices that we've been testing so far include a tablet device with a macro lens. And the idea here is tablets are flat. This is something that could be easily packed out. And it is something that could also be used to collect other types of information in the field if needed. The other preliminary device that we've been looking at is a point and shoot, the OM system TG7. We're drawn to this for a number of reasons, including the fact that it is waterproof, and they also say shockproof, and the size. So overall, this photography approach, like with this, the point and shoot camera, there's all kinds of different things to think about, but I absolutely loved Kylie's creativity here. Instead of thinking about a stand necessarily, what about a soil knife and how could this be standardized, right? So there's a lot of thought and discussion going into this, but we're definitely in the middle of field testing and looking at some of these preliminary approaches. And then Cassie had the opportunity to test the tablet approach up in Southeast Alaska during a recent multi-week trip for field work. And you may be thinking, oh, yeah, it's really nice to test this in the nice sunny day that Cassie encountered up in Alaska. But Cassie also had the opportunity to test this under more realistic conditions within the rainforest system. So we're definitely thinking about weather in this context. So overall, as I wrap up today, these are just some of the preliminary images that we've collected using these devices. And we're also comparing it to a more, Mark II camera that you could set up in the lab for consistent lab-based photographs. And the next steps for this photography protocol include a lot more field testing and standardization, images, PED selection, even the area of interest selection, and how to have this consistent across the board. And then Kylie is also working on this automated image analysis technique, thinking about Root Painter or other types of software that could be applicable to try to automate the process once you've actually identified root distribution on some of these samples. Kylie's working through the process of what segmentation would look like, and most importantly, to be able to generate enough images to create some training data sets and potentially even a full model where you could load in photos, maybe for the coastal temperate rainforest as one example, and then be able to calculate out or determine the distribution of roots as one type of example. As you can imagine, lots of potential limitations, and this is, again, just the kickoff of this work. We're really excited about it, but we also understand that there's a lot of limitations and things to work through. So overall, to wrap up today, we have taken a cross-scale journey thinking about the cross-scale analysis of soils and what you can learn about soils across landscapes or across lithosequences, and then going down to the microscale of study, where we've been thinking about co-location and combining different types of high-resolution imaging techniques, as well as the use of micro-XCT for analyzing grains and potentially visualizing soil fungi. And then finally, thinking more and more about fungal adherence and what this means, and how there are different types of fungal grain associations in samples, and what can we do to categorize these and learn from them. And finally, bringing it back full circle to thinking about how we can quantitatively collect data in the field, especially from a biological soil health indicator side. So with that, I just want to thank you all. I've got my email address here. I'm also going to stay online, so happy to answer any questions in chat as we wrap up with Q&A.
Okay, thank you, Rebecca. Appreciate your seminar. Wonderful survey of a lot of work that you're doing. So right now, I don't see any questions in the Q&A. Again, if you have any questions and you want to put them in either the chat or the Q&A, put them in the Q&A. I think that would be a little bit easier to keep track of. But for now, does anybody have a question? It looks like Skye, do you have your hand up?
I do. I have a quick question to get us started.
Great.
So in this environment, we are always being asked, so what, who cares? So what's maybe your grad students want to take a swipe at this. What's the who cares? Why? What can we tell our funders and supporters when they ask us, why do we care about this fundamental research?
The students can definitely jump in. So Cassie, Maddie, or Kylie, you can jump in. On my side, with respect to approaching funders on this, I have to say it completely differs on the government agency, right? So the Department of Energy is investing a lot of money in this area of research right now, and it's all about understanding nutrient cycling and controls on nutrient cycling, especially from an incipient weathering standpoint. So again, this enhanced basalt weathering work, these types of applications. And most recently, there is a big push on the DOE side for critical minerals and being able to understand the microbial controls on what would allow critical minerals to become more available or, obtainable in the environmental system. That completely differs when you go to thinking about NASA, right? And if you're going to NASA and thinking about mineral weathering, it's all about biosignatures and being able to understand both what may be evidence of life that could be preserved for long enough timescales to be detectable on Mars or other systems, but also being able to detect false positives for life, abiotic water-rock interactions that may lead, to, if this is something that can be biologically produced too, everyone's excited, oh, we may have found evidence for life. Oh, wait, water-rock interactions produces that same exact type of feature. So big picture, I'd say it depends on the agency that you're going to, but there is a lot of emphasis on this. You just have to kind of know how to present it and the specifics of what that agency is looking for. But Cassie, Maddie, or Kylie, do you want to jump in with any more information?
Well, I think for the field photography protocol, I feel that it's opening up new avenues for understanding soils to more people. If we can make it, simple enough and easy enough to, have other people dive deeper into fungi and understand their role in ecosystems on a visual scale, I think that's important for me.
Great point, Kylie. Yes.
Okay,
I also saw a quick question in chat from Scott. Hi, Scott,
Yes.
by the way. So the question is what particle sizes you use for the mineral weathering mesh bag experiments. And the particle size range was between 250 to 53 microns. So that was, the idea is not to have it so powderized that you've just got like basaltic powder that may not even stay within the mesh bags necessarily. But then also that it's not so coarse that you may not be able to detect any type of incipient weathering after only a couple of years. So that is kind of the range that we used. And it was also part of a broader collaboration with a colleague of ours who actually used the same kind of rock material in coarser sizes for a lab-based experiment. And so his particle size range was higher, but we had this subset of material that we were able to prep and use. So it was kind of like multifaceted, but the scientific justification for that was just, again, not to have powderized material, so that 53 micron cutoff and not have it be too coarse grained.
So there's another question in the Q&A, Rebecca, from Faith Lewis. I'll go ahead and read the question for the sake of time, but do you plan on attempting to identify the fungi associated with particular minerals and or associations in the future?
Yes. So we are so excited to have a new collaboration kicking off with Dr. Aaron Nuccio at Lawrence Livermore National Lab. And this has been one of the ongoing questions. And I'm not a microbial ecologist, so I've been looking for the perfect collaboration in this way. And Aaron has been a fantastic collaborator. And we're basically, the idea is to be able to have these samples and to say who they are, and maybe even trying to get at some of the function. Maddie, do you want to jump in and talk just very briefly about your current project with Aaron and how it relates to this question about identifying the fungi?
Sure. Yeah, we're starting a new project. It's actually ongoing. The plants are growing, in which we have 2 different tree species from the southeastern Alaskan rainforest planted in field soil. Yes, we did transport a few hundred pounds of soil from our Southeast Alaskan field site. And the plan for this experiment is to use the 13C isotope of carbon to label both the plant material, and then as the carbon is allocated to the fungi, we can track that 13C marker into the plant-associated fungi, which can be used in a specific sequencing method to identify those particular fungi. So our goal is to look at the different species composition and functions in the fungi in these experimental treatments. And we also have mesh bags, kind of like Rebecca explained, inserted inside the pots. So we would also be looking at mineral-specific effects on the microbial communities.
Thank you, Maddie.
Hey, fantastic. I think for the sake of time, we're going to go ahead and move on. But if, and by the way, Rebecca, I have just so many questions and comments and thoughts I want to chat with you about. This is really great presentation. Brought up a lot of thoughts. So I'll reach out to you separately, and I encourage everybody else on the call to also do that. You can do that in the chat, even while our second presenter is presenting, but so we can maybe address some things at the very end. But for now, I want to introduce our next speaker. It's my pleasure to introduce Markus Steffens to you. Markus is a soil scientist and group leader of the Soil Fertility and Climate at the Research Institute of Organic Agriculture in FiBL in Switzerland. He's a lecturer for soil science at the Institute of Geography and Oeschger Center for Climate Sciences at the University of Bern, Switzerland. His research is on soil organic matter, infrared spectroscopy, and sustainable soil management and farmer-focused dissemination. And he works on various national and European projects at the interface of soils and climate-smart agriculture. I got to meet Markus some years ago at a conference that Alfred Hardemink put together in Madison, and afterwards was able to spend a little bit of time with Markus and my oldest daughter, who at the time was like 12 years old, is now graduated high school. But we went and visited Markus while he was still at the University of the Technical University of Munich, and that was a great time to meet him, and was a wonderful host. So I'm excited to hear his talk and everything he's been up to since we had our last visit. So Markus, feel free to take it away.
Hello and good evening. So I'm in Switzerland, and for us it's 9:30, so it's a little bit dark. You also see it, and I'm thrilled that Daniel invited me, and I'm really happy to present the work that we are doing since 2010, more or less, and it started at the Technical University of Munich. I start sharing the presentation. Do you see it?
Yep, we can see it. Perfect. Thanks,
Great.
Markus.
Yeah, so it's about imaging spectroscopy. And as Rebecca also introduced before, of course, this is not just my work, but many people were participating and were part of this work. And it started in, yeah, as I said, 2010 in Munich, or in Freising, that's close to Munich. And then I collaborated also with my university where I studied in Germany too. So it's in Trier, and there it was the remote sensing department. And afterwards, when I moved to Switzerland, I continued also working at the University of Bern with this one. So generally, my main, so 8% of my working time is at the FiBL. So there is more applied research and dissemination of research to farmers in EU. But I still try to keep a foot in the research, in the basic research. But I think I find it very interesting to have this combination. So to see on the one side the application in agriculture, and on the other side also the basic science and how we can combine this one. And I think it's a very important thing to also disseminate and communicate the results that we have, and of course also try to scale it. Okay, so many people, maybe you have seen, Daniel is also on it. So we started a collaboration, but in the end we both moved. And so we did not come to the point that we finalized, or that we really got into the details. But maybe we have some years left. Okay, so soil science today, mostly we are talking about horizons. And of course, we always think that, we have to assume that the horizons are homogeneous. At least that's the approach of the classification system, which we are all using. But in reality, I think all of you know soils look more like this. So they're far away from being homogeneous. It's more the question, where's the heterogeneity so big that we can call it a new horizon? And so for me, it was I wanted to have a technique to better represent, or to better measure, this heterogeneity in the field on the Pedon scale. And so we have in agriculture, in soil science and in geoscience in general, we have various spatial scales. Of course, we are talking about landscapes, heterogeneity or variability. We talk about Pedon variability and also aggregate variability. Nowadays, and of course, biological, chemical, and physical properties, not just one. Everything is changing on various scales and on orders of magnitude. Also the biological or the parameter properties in general, they can have very sharp differences between different scales and different positions. And in the end, also the properties and mechanisms that lead to these properties and to given soil types, they are intertwined across different scales. Nowadays, we have remote sensing. This picture is that's a scheme of a German satellite which was launched in 2017, I think. So it's more looking like a washing machine, but that's the NMAP, an environmental mapper. And my friend with whom I did this imaging spectroscopy part, he is a remote sensing scientist, and he was on the core science team of the satellite. And this is also the reason why this whole work started, because he was responsible for this hyperspectral camera. And in the same direction as Rebecca was working in Freising, we were also working on the aggregate scale. We were using nanosims. But in this talk, I'm not going to focus on the nanosims. I just recently added a last slide. So if there are some questions, or maybe I can show it at the end, so to make the connection to Rebecca too. But now I'm going to focus more on the Pedon scale. And I think that the imaging with NIR spectroscopy is a good technique to record and to cover the spatial variability on the Pedon scale. In the end, how do we do it? The camera is either flying on a satellite or in the plane, or it's standing in the lab for calibration. So we need to bring the soils to the lab. So we were thinking about using the camera in the field, but that was not too complicated, but we were more on the proof of concept phase. So we wanted to try it in the lab. And for this, we built a box that was in 2010. So it was not that normal to have a soil auger with inline cores and driven with a pneumatic auger. So we built it our own, and we built a stainless steel box with a dimension of 30 by 10 by 10 centimeters. And this was hammered vertically in the soil. And then you just excavate it. And you see it lying here now in the low part. Maybe you see my mouse. So you'll see it lying here. And this camera, this black box, that's the camera. In this case, it's a HiSpecs. It's from Norsk Elektro-Optik. That's a Norwegian company, but nowadays, of course, there are others. And this one is covering, this camera that you see here is covering the spectrum from 400 to 1000 nanometers. We used the second camera, which is sitting directly beside this one, and this is covering the spectra from 1000 to 2500 nanometers, so that we can cover the whole visible light and near infrared part. Now, the interesting thing is that it's a camera, so it's not just one measurement, but in the end you have 1600 detectors sitting beside each other, and each one is taking one point. So that means we have 1600 pixels, and we can measure as many lines as we like. So it's called a push broom scanner. So it takes a picture of a surface, and then you move the surface. Normally it's flying on a satellite or on a plane, and so it's flying across the surface. In this case, for calibration, it's put on the translation stage, so the sample is moving below the camera. And with this setting, so there is a lens inside, and each pixel has a spatial resolution of 63 by 63 square micrometers. So it's quite small pixels, so a very high, not a very high, but a good resolution. And for these cores, which we took, so 30 centimeters long and 10 centimeters wide, these were more than 7 million spectra or pixels per image. And what we did, so we took one picture, then we sampled some interesting spots, we planished the surface again and took the next one. So in this case, that was the very first core which we took. We took 7 images. So in the end, we got not a full volume, but we had 30 centimeters long, 10 centimeter wide, 10 centimeter deep. And for this one, we had 7 images. Now this is illustration, but when moving how the measurement was done, but when moving it to the newest version of PowerPoint, it completely distorted my presentation. As you can see here now, it should have been the box and it should illustrate it. So in the end, this is illustrating what we did. So it was scanned, we took samples at these spots, then we planished it, scanned it again, and in the end, now that's the result. So we got 7 pictures. So here you see how this recording session, the first one, developed. The other 7 cores. And to make it even more clear, so on the right side, you see the image of the core. And for each of the pixels which you see, you get a full spectrum from 400, in this case, to 1000 nanometers or to 2500. Now to go a little bit more into the details. So the first one was we tried if it's really working. So that was the proof of concept. And so we just went to the next forest close to Munich. It's a löss soil, so it's the Stagnicluvisol in the WRB or in the Endo-Aqualf in the soil taxonomy. And we just want to try and see and play around with the data and see what we can do. So here you see a real color image now. That's nothing surprising. But then we did geostatistics. And what you see here now is, in the end, it's the ZIL for the people who are used to geostatistics. This is showing how different a pixel or a region is to its surrounding. And so the more reddish it gets, the more different it is compared to its surrounding. And the more bluish it gets, this pixel is very similar to its surroundings. So you see here, for example, that's a region where it's very homogeneous, and the red parts, this is very heterogeneous or very different particle or substance. And this was the very first thing that we did. And I think it's a quite nice tool to identify hotspots or to see where is a difference. Because if I have to sample a soil profile, I don't know where to take something, if I'm not trained, or if I don't know what to look for. And this gives a first indication where are spots which are very different to its surrounding. The next step, of course, was the most, I think the most clearest one, which I was looking for, and that's classification. So you do a supervised classification. So you tell the computer or the program, this is an AH horizon, or this is a BT, so a clay enriched horizon. And then you can use classification algorithms, and they tell for each pixel, is it which type of horizon it is. And this was done in 2010, 2011. And nowadays, of course, with AI, it's all these classification things are getting much, much more better. But still, I think also at this point, it was already nice. So in the end, for me, that's a proof that we can do quantitative soil classification. So if you remember this introduction slide, how most horizons look like when we describe them, with this one, we can get a quantitative soil classification. So how much area is covered by which horizon. And the third step was, of course, we took samples and we did classic analysis. So for carbon, nitrogen, and then for some metals, so aluminum, iron, and manganese. And then you can imagine you can do regression analysis, and then you can make maps, so elemental mapping and soil profiles. And now here you see carbon, nitrogen, iron, and manganese. Of course, this is also the very first try. And in the end, there's, of course, potential for improving, but I think it's a quite good proof that we can do elemental mapping on continuous soil profiles with a very high spatial resolution. And you can see, for example, here, this feature here is a root that we identified, or you see also this separation of the reducing and oxidizing parts of the B horizons. This was done now for 10 centimeter wide and 30 centimeter long cores or boxes. And with a project in Germany, we could also take inline cores. So this was then taking place from 2015 on. It's nothing different than the other one, but we just could scan cores. So we have intact soil cores of 1 meter length. And here you see now 5 cores, which we took. That, as I said, it was a project, was called Bonares in Germany. And yeah, then you see some intermediate results. So these are principal component analysis. So these are the next 5 colorful pictures. Basically, it's just showing where you see differences, where are different materials. And then the last 5 that you see here, this is the carbon map. And we again see quite nicely the AP, so the plowed topsoil horizon, which is rich in carbon, which is the blue here. And then you see this very sharp edge where you have the plow pen, or where the plowing stopped, and then you have less carbon. Other features that appear is this structure or this feature here, which is either an old root channel or it's from an earthworm burrow. And the last one here, there you see the variability of the carbon with the depth. And of course, as you can imagine, the carbon is quite homogeneously distributed in the AP horizon, but it's getting much more heterogeneous and variable in the lower parts. Then a second step was, because we have seen that we see differences in the organic matter. And so we said, okay, let's try if we can also separate different types of organic materials. And for this, we went to the Alps, to the German Alps. And there was an area where a storm was going through, and the forest was taken away. And now we had a bare area where the thick organic surface layers were fastly decomposing. And so we wanted to make a comparison of the organic surface layers in the forest and beside the forest. In the end, we took several cores, but we analyzed just one, as you see here. This is how it looks like. This is not totally 30 centimeters because it's quite loose. So we lost a little bit of material. So it's 25 centimeters long. And we could not take 7 or more slices of these blocks, but just 3 because it's so loose, and it's purely organic. So that's just organic material from the forest floor. And what we then did was the same again. So we took samples and analyzed them for carbon and nitrogen, and we used NMR to get information about the chemical composition of the organic matter. Here you see again, that's the principal component analysis again, and we see in this just organic surface layer that there are different types of organic matter. And maybe you already recognize that there are some fresh roots, for example, and these roots show other colors, so other principal components, other spectral bands which are active when compared to this more thick materials, for example. And then we did unsupervised classifications, and we got this result. So that's not supervised, but unsupervised, just based on the variability of the spectra. And from the first indication, it looks good. So we have, for example, this fresh root. It's green, and everything in bright green, that's living or very fresh organic matter. And then we have 2 different other compartments. And so we used the NMR. So here you see a close-up. And so class 1 is the living roots and the undecomposed organic matter. And then we have class 2, that's particulate organic matter with a high lignin content. That's this one. And here you see the results of the NMR. So we analyzed the samples that we took, and we see that these areas are high in lignin, so in woody materials. Of course, we're in the forest. And this is typical for the second class. And the third one is the amorphous organic matter, which is highly decomposed. That's the remaining part. So that means we can, with this imaging spectroscopy combined, of course, with digital image processing, we could separate without any knowledge. So that's not supervised classification. It's just unsupervised classification. We could separate different types of organic matter in a purely organic surface layer. Okay, and with this information, so we have this first proof of concept, then we have this study on the different types of organic matter. We said, okay, now go back to the mineral soils again and see if we can quantify different types or fractions of organic matter. And so I did my PhD in Germany, but about soils in China, about overgrazed areas in northern China. And I revisited this area. So you see it's north of Beijing. It's a calcic chernozem, so in the vast steppe areas close to the border of the People's Republic of Mongolia. And there is a shortgrass steppe, and we had access to grazing experiments or grazing exclosure experiments. Yeah, these are the soils, and we took again some cores. And in this case, as I said, grazing experiments, we knew, okay, on the left, we have an ungrazed area with a lot of organic matter. On the right side, we have a grazed area with less organic matter, and particulate organic matter especially. And so we want to see if we can quantify it. And for this, we split. So we have the 2 cores, and we took, again, layers in this core. So it's not vertically, but it's going back, you know what I mean? And we sampled these small squares and fractionated them. So we reduced the size of the square so much that we can just do 1 fractionation per square. And then we did a classic physical fractionation, which we were good at in Freising. So density fractionation followed by a particle size fractionation. So we get the F-bomb, the O-bomb, and mineral associated organic matter. And for each of these squares, now we got the numbers, how much is inside. And then we did our normal digital image processing. And you see again, so in green, we get again the fresh organic matter. And we see that the correlation was quite good. And then we continued with this work. And in the end, we could show that we have a good correlation between the physical POM, particulate organic matter, which was extracted in the lab, and the spec POM, so the spectroscopic POM. So in the end, that was the proof. Of course, that's not one-to-one line, but we are quite close to predict or to measure the particulate organic matter content in the undisturbed soil profiles with spectroscopy. This graph on the lower right, you see that the scale is not linear. So the y-axis is not linear, but that's because of the particle size. In the uppermore horizons or boxes where we fraction it, there are very big particles, and they make it quite complicated to quantify the POM correctly. But still, we got quite good representation with the spectroscopic POM. Then for the last, or not for the last, for the next one, we went again close to Freising or to Munich, and we sampled. Oh, there was something. The writing is behind the maps, but it's not that important. That's very close to our to the lab where I was, and there was the idea. So here is a rectangle, or not a rectangle, it's a triangle with a beech forest, with a beech stand, and directly beside it is a stand with a Douglas fir. So, and we know from mapping with students, so mapping courses, that the soils are slightly different. So in the beech you have a luvisol, and in the Douglas fir you have a stagnosol. So just beside each other you have two different soil types. We are quite sure that it's not because of the beech of the trees or of the species, but it's more because there's a construction site. So you see that there's a shooting range, and when constructing, they most likely compacted the site where the Douglas fir was planted, and there you have the stagnosol. So our idea was now to have several cores from this area and to see if we can use our technique to, or if the technique is able to separate the different soil types without our without the knowledge of us. So we did the same thing again. So we did classifications, we took samples, so we measured carbon content, and we measured especially iron and manganese, because we were talking about, or we were looking for luvisol and stagnosol, so reducing oxidizing conditions and aggregation, and how the iron was redistributed. And now you see that's an animation. So that's the result of the classification. On the left side, you see the three profiles from the beech stand, so the luvisol, and on the right side you see the three cores from the Douglas fir, which is the stagnosol. And the greenish colors, the brownish colors are clear. This is the humic A horizon, and the greenish colors, this is the typical luvisol horizons. And in blue, these are the stagnic horizons, so where you have reducing and oxidizing conditions, and where you have the redistribution of the iron and manganese oxides. And what we see is that the algorithm, or not the algorithm, but our approach was able to separate these two soil types. And this is still ongoing. So we started the project since several years ago. Then I moved to Switzerland, and it was in the not on the desk, but it was below the desk. But now we are looking at the data again, and we are now working on it and trying to get it published. And we are quite happy because we can separate these very close or neighboring soil cores and use imaging spectroscopy to separate different soil types and quantify the different diagnostic horizons. And now moving a little bit away from the classification, because when doing this one, we were also in another project, another German project with the UFZ, which is specialized in soil physics. And we thought about combining X-ray microtomography with our imaging spectroscopy. So we wanted to see if we can combine physical characterization with this more chemical characterization, what we are doing. And for this, we went to, it's a pity now I don't have this introduction with the soil type and so on, because I did not find it. This is coming from, the soil was coming from close to Cologne, where you have open pit mining for lignite. And we took samples from recultivation sites. So we had a chromosequence from freshly deposited material to 25 years old, which was again used for agriculture. And so the project was looking if it's possible to recultivate the soils and how fast aggregation and structure develops and where the carbon is going. This was the project, and this what we took some of the cores, because they were scanned first for microtomography, what you see here, which is the pore distribution. And then we took slices. So you see these planes in the scheme, and these slices, these are the layers where we took hyperspectral imaging or imaging spectroscopy images. And you already see, especially for here at this point, for example, where you have the root, that the surrounding area around the root has a different color than the rest. So in the end, we tried to see if we can combine these two techniques. And here are the results. So you have different slices, and you have what you see here now is our classification. So we could classify voids or aggregates. No, not aggregates, sorry, voids. These are pores, sorry. We could characterize organic matter-dominated areas, manganese-dominated materials, the pyrolefs material, and iron oxide-dominated things. So we could separate them in the image, and then we could combine them with the microtomography results, where we can show which of these points is connected to a pore and how far the next pore is. Yeah, so to sum up our approach, I called it 10 years ago, it was the soil imaging with NIR spectroscopy, which is a quantitative soil science on Pedon scale. So we can use it for doing more quantitative soil science. And the first one was a proof of concept. So we could show that we can map spatial heterogeneity or spatial variability. We can do quantitative soil classification. And we can do elemental mapping. Everything with a spatial resolution of 63 by 63 micrometers. Just to add, you can also add another lens to the camera, and then you can go down to 20 by 20 micrometers even. But then, of course, the width is reduced, so we could not take these big cores. Then we could show that we can map soil organic matter quality, so different types of organic matter, and we could even quantify different organic matter fractions. So fractions which are really physically fractionated in the lab, and we can quantify them with the camera. So we have also something like a spectral POM, particulate organic matter. We at least made a step towards fusing 2D and 3D techniques, so our imaging with the microtomography. And we could also use this one not just on these boxes, but also on long cores. So up to 1 meter is possible with this with this lab rack. Of course, you can scan the next one, so you can do it in meter parts. Yeah, so I think it worked quite well. And now, since I moved to Switzerland, now we are in several EU projects, and we have one which started last year, which is called Deep Horizon. It's called Deploying Ecosystem Solution to Improve Soil Health and Uncovering Subsoil Functions in the Critical Zone. And here on the left, you see the soil pitch, which we sampled 2 months ago, not far from here, from our house. And this is a cambisol on a moraine. And in the project, we do, again, hyperspectral imaging. So we scan cores of these profiles, and colleagues do a lot of analysis on carbon or on chemical, physical, and biological properties or parameters, and we will try to find them in the cores. And another project just recently started in September, which is called CAFAMOR, which is about carbon farming monitoring and registry. And there we are also doing imaging spectroscopy. In this case, we want to better understand the spatial heterogeneity and variability of carbon in the first 50 centimeters. So we are scanning only 50 centimeter long cores, but we want to understand how the carbon is redistributing. And in the sense or in the context of carbon farming, of course, when we have compensation payments, we want to understand how the carbon is distributed and how different measures, agricultural measures, change the distribution, not just plowing, but also different crops and crop rotations, and how fast we can measure differences. With this, of course, we want to feed models or to get a better understanding, because in the end, the carbon farming and all precision agriculture will rely more and more on Earth observation. But Earth observation, of course, or remote sensing, gives only results from the first micrometers. And if we have a better understanding of the dynamics, temporal, so the spatial variability, which I said before, and we will measure it quite often in the year. So we will want to have better understanding of the spatial and temporal variability of the carbon. And for this, we are going to use hyperspectral imaging again. Thank you very much.
Thank you, Markus. Fantastic work. So much to, I'm sure, so much to sink our teeth into. So, all right. Do we have any questions? If you do have any questions, please feel free to put them into the Q&A. And while we're waiting here, I have a question, Markus, about the unsupervised classification. You showed that there was essentially it was being. How does that work? How does the unsupervised classification work? Is it a k-means type clustering?
Yeah. It was a k. Of course, it was a little bit more complicated, but it's based on k-means. It's k-means. But of course, we first had to get rid of shades, for example, and surface roughness. I did not go into detail on this one. I think we started talking when we met first and second time. Because one point is we can work with a surface roughness of a few millimeters. But of course, as soon as you have more roughness, then you have more shades. And in the shades, you have less good spectra, and so the classification gets more complicated. And so the first steps we tried to get rid of shades. So the first k-mean clustering steps, you got a lot of classes with shades inside. And so first we had to do some classifications to remove shades, and then had to reclassify again after taking the shades out. And so, yeah, it was quite complicated and work intense. But it's based on k-means, yes.
Okay, fantastic. And also, you mentioned that you could go down finer resolution if you used a different lens, but the problem there is that the field of view decreases as you scan. I'm curious, have you had any luck or tried running essentially multiple scans with some overlap so that you could go wider at a higher resolution?
No,
so that you could stitch the scans together.
Sorry?
Oh, just thinking about how if you could stitch the scans together.
Yeah, you can. That's so. At FiBL, I have a new postdoc, and she's very good in coding, and she started working, or she did her PhD on imaging, not imaging, on just spectroscopy, and she's now playing around with the camera, and she's doing exactly this. But at this time, we had only access to the 30-centimeter lens, so we could only work with this one. And if you would go widen the field of view, the image would not have been sharp. Would not have been at the right focal plane.
Right.
So we would have to buy a new lens. And this at the beginning, this was just playing around with the instruments. So we had no budget at the beginning. So the first 3 publications were virtually without money. I never thought about it, but it was just evenings
labor of love.
Some beers with a friend.
Yeah. Excellent. Other questions?
I had a quick question.
Yeah.
Markus, amazing presentation. Wow, I am so impressed, and it was amazing to see how you're working through being able to separate out the different types of organic matter in these field-based scans. I was just curious, could you walk through a little bit more on what it takes to prep one of these profiles to be able to scan, like in terms of field logistics and conditions, and even are there certain types of profiles or soil properties that are more suited to this or others that are more challenging? I was just curious about the logistics.
In the end, so we never had problematic things. So if you have a lot of rocks, of course, it's getting complicated. And if it's too loose, it can fall out. but we improved this one. So in the end, the first boxes were just built by my dad, because he's a welder. And so he built me the boxes. And the first one, of course, it was too thin. So we had to build use thicker walls. But then it was getting too thick, and it was very hard to drive it in the soil. And so it depends on the soil type and how you can do it. In the end, it's the same as with if you do resin profiles, it's going in the same direction. But this was more the starting point. Now we are using a line, so soil augers, pneumatic soil augers with liners inside. And this is no problem. So I had this one picture illustration. Wait. No, where is it? Should be close. This one. And we were quite happy with these cores. And this is also done by people in geology. So sedimentologists, they are quite used to take meters and dozens of meters of cores, and you can scan all of them. And it's quite likely that people in geology also have cameras around for core scanning, and also for ice cores, for example. So it's used quite frequently. But these people are not interested in soils, because soils are dirty and complicated, and so they are sticking to minerals and rocks. But you can also use it for soils.
Awesome, thank you.
You can also drill, of course.
Yeah, that's great. Just a follow-on comment on that. We've been taking monoliths from the pit face. Markus, I know you and I have talked about that a little bit, but we use a diamond bit saw, a cutoff saw, and we just cut it from the edge of, or the face of a profile, and then are able to extract it that way. So we haven't gotten into really hard rock yet, but we have gone through some pretty hard petrocalcic nodules, and we can just slice right through them with the diamond saw. And so we're able to, maintain the intact structure, even with more problematic type soils. So, yeah.
We were also thinking about using it in the field, so we could also, with my friend, he was easy. We could have also tried to take it to the field to scan it directly in the profile. And I was also writing to a federal office in Germany for having a patent on this one. But so you have a scaffold that you put in the profile and where you scan it, because you have to maintain the distance between the face and the camera all the time. But they said, no, that's nothing special. You can buy it everywhere. And so we did not get money, and so we did not follow it. And the other point, sorry, I don't want to put it too long, but Rafael Vizcarra-Rossell, so he's in Perth, in Australia, and they built a trailer in the end. And in this trailer, they drill a core, and it's taken out automatically, and then it's scanned for half an hour or something in the trailer, and they do whatever you can imagine. So they are doing, I'm not sure if they are doing the hyperspectral imaging, but they are doing XRF and I think X-ray, no, not X-ray, gamma ray, I think it is. And so you get many, many readings for these cores, and they did this also for mapping. I can send also around, he has a publication, 2018 or something like this.
Excellent. Yeah, thanks. If you get that to me, we can get it out to the group. Yeah. Perfect. Okay, I'm not seeing too many questions, but I have just I guess one more quick one about the nitrogen. I saw that you had nitrogen mapped on there too, next to the carbon, and I'm curious, is that nitrogen? It didn't look like it was immediately correlated to the carbon. Is the spectral response able to essentially pick up the nitrogen over the carbon, or is it really just kind of picking up some kind of organic carbon or organic matter type signal?
No, I think it's just because of the carbon. So it's the CN ratio is quite stable, and you get it about via the carbon. We were thinking about using dyeing agents, so something to put a chemical which is dyeing depending on the nitrogen content. Of course, this is only the total nitrogen which we get from the elemental analyzer. So we did not analyze for mineral nitrogen or something like this. So in the end, it's just by correlation.
Okay. Well, fantastic work. I'm not seeing any other questions here, so I think I'll well, actually, sorry, there is a question from Catherine. Very cool presentation. Thought the mapping of SOM fractions was particularly exciting. Do you think that this is a technique that could be applied at scale for soil survey? Seems like it would be a much more efficient way to quantify SOM fractions than traditional density separation methods.
I think so, yes. But of course, we have to see you need calibration. So we did it just on these two soils, which were directly beside each other. And of course, it depends on the distribution within the core and in the soil profile. So you could also have points where you have a lot of particulate organic matter. So for like earthworm casts or burrows. So you would have to take several slices but I'm sure that you have to have a more or less good representation. And then, of course, it's the question, is a supervised or unsupervised classification sufficient, or should we more go for spectral libraries? That would, of course, we did not do it in this one. Now for this new project, we are working on this one. So we have a lot of infrared spectral libraries for different materials, very, very many different materials. And we could use the spectral libraries to map different types of organic matter and also different minerals in these images. So what we did was just this image without any other library data. And in Germany, you have the Lucas data, and I think you have, so for soil monitoring, so they have more than 100,000, I think more than 200 more or less samples now, which are being scanned. And I think in the US, you also have big libraries of infrared spectroscopy data, and you could use them, of course. And I think it's a good approach for mapping also. Maybe you can, especially when you take these cores, which is quite fast, and you can scan the cores. And when the scan once is done, you can also take samples and destroy the core because you have the image, and then you can analyze it. And I think it would be a good technique and approach.
Yeah, it's great. Thank you again for your presentation. So I know we're a little bit over time, so I want to go ahead and conclude our webinar today. Just really thank everybody who's attended, and my understanding is that this is recorded and will be posted somewhere online. Skye, can you speak to that for a second?
Yes, it'll be recorded. Everyone who registered will get a link, and then it will also be posted to the National Cooperative Soil Survey YouTube channel as soon as we get the transcripts edited and finalized.
Great. And I know that we're planning on keeping up this webinar as we go forward. So be on the lookout for the announcement for our next webinar series. Thanks again to our speakers and to our audience, and hope everybody has a great rest of the day. Thanks.